and welcome everybody to our 219th episode. My name is Benji Cohen with the Fish and Wildlife Division of the Minnesota DNR, and we got a really fun program today. I get to working with Craig Superior. He's what he used to be. Now you're the Southern Region Fisheries Manager, right?
That's correct. Yep.
Great. And I learned a little bit about this during the fishing open. We were down there in Mankato a couple years ago, and it's kind of a really cool program. So I'm excited to have you on here and share with everybody a little bit more about the lower Mississippi strain of walleye we've been working on.
Yeah. Well, thank you. Thanks for having me today. I'm really excited to be part of this Moss webinar. Um, And so I'm Craig Superior. I'm the Regional Fisheries Manager for the Southern Region in the Minnesota Department of Natural Resources. I'm going to be talking a lot about the Waterville area in here, so I apologize if I say my area, but I used to be the Waterville area fisheries supervisor up until this February when I took this job. So, but this project, this Lower Mississippi River Strain Walleye Performance and Reproduction in Southern Minnesota, is kind of a project that formed a few years ago, and it's a really interesting one. It's had a lot of consequences on our walleye management, and especially down in this part of the state, but also statewide. And so it'd be a really good one for you all to hear. And a lot of the locals down here in the Mankato, Waterville area have heard this a lot over the last couple of years because we've been championing this project, and I'm excited to be able to provide it to you today. So. Uh, Let's go in here. All right, so walleye stocking in Minnesota dates back to the early 1900s. Actually, 1910 was the earliest stocking that we have on record. And we literally used to stock fish that way, which is shocking. We used to haul around fish in rail cars, and we'd have milk jugs in there, and we keep them on, keep them cool, chilled, and we'd walk up the shore and dump them out in our lakes and anywhere we were doing stocking. Don't necessarily do that so much that way anymore, and we also don't necessarily wear suits when we're out in the field anymore. If you see the DNR staff out, but stocking goes way back to the 1910s. And the thing about stocking walleyes is, when we first started doing walleye stocking, we really didn't consider genetics as an important part of a consideration for stocking. And until about the 1980s, 1990s, we really started looking at genetics on a statewide basis because technologies had advanced to the point where we could actually assess genetics. And we started considering this in our stocking programs for walleye and other species as well. But some of the research that was originally done looked at what were the genetic composition of our fish in the state. And what we found was that there was unique genetics distributed throughout the state in certain areas. And we started to develop specific genetic conservation units, because what we didn't want to do is take fish outside of one genetic conservation unit and stock them in another genetic conservation unit and risk having those genetic strains, maybe be converge or take away their uniqueness to that area. So that's why these genetic conservation units in fisheries really makes a difference. But what we found was that we had three three really unique genetic conservation areas in the state. The red dots kind of show where we had egg take stations historically within these units. But one of the things I wanted to point out to you is that if you look at the southern part of the state, we didn't have a really name for it. We called it the universal stocking zone. And at the time, we didn't think there was a lot of natural reproduction in the southern part of the state. The lakes in this part of the state they're impaired. We have a lot of agriculture and other watershed disruptions that have caused issues in our lakes with water quality and other things. And so we just kind of assumed there wasn't a lot of natural reproduction. So that being the case, we could take walleye from any part of the state, bring them down here, and we'd be safe because there's nothing really native to that area that we'd have to worry about disrupting. So we had this kind of universal zone in the southern part of the state that we were operating on for a long, long time in Minnesota. Let's see. All right. And then along come something we found really starting in the late 1990s and early 2000s, was this lake down in Murray County in southwestern Minnesota, Lake Sarah. And this lake had a very unique population that started in the 1990s. They stopped stocking it altogether because it was naturally reproducing. And one of the few lakes in the entire part of the state that we didn't have to worry about stocking because it was just naturally producing, and we didn't really know why or what the reason was. And so this is a little video that we got from our Wyndham Fisheries office. I'm going to play it here if I can get it to work.
We go around and we lift the nets and dip out our walleyes. And we're really looking for our ripe females as well as your males that are ready to spawn as well. And then we bring them back to our trailer here, and then we go through the process of actually stripping those walleyes eggs out of these fish. Once we get, basically 2 males and a female into a pan, then we augment it with the water and activates those micropiles to open up so that they can be fertilized. And then we typically stir them for about 2 minutes. And once that 2 minutes is up, then we add bentonite to the pan to keep the eggs from sticking together. Once we do that, we put them in a cooler and then we give them a little ride over to the Waterbill hatchery. Lake Sarah is a population of fish that has consistent natural reproduction. We haven't stocked it since 1991. It's our best walleye fishery in our area. We had these fish genetically tested and found out their unique genetic strain, alluding to the fact that maybe that is one of the reasons that we have natural reproduction. The eventual hope here is that we can create other Lake Sarahs in southwest Minnesota, with the idea that from a fish management standpoint, the ideal walleye population is one that actually takes care of itself. You don't have to do maintenance stocking or to augment the population. It does it on its own.
So what we found out through this process of taking eggs down in Lake Sarah in southwest Minnesota is that we had this unique genetic strain that we really didn't know about because it was naturally reproducing. And so the Lord Miller, our fisheries geneticist, was studying this, and he took some of the genetic tissue from the fish down there and kind of analyzed it and looked how it compared to the rest of our state as far as the walleye genetics. And if you look at this particular figure, this kind of shows a genetic footprint of our walleye in Minnesota. And we talked about the genetic conservation units earlier, the genetic management units. You can see those kind of played out here, those 3 or 4 units. And historically, we had been taking a lot of our eggs from the northern egg take stations at Cutfoot, particularly in Pine River and Brainerd, and this one's up at Grand Rapids, and taking those fish and bringing them down to the southern part of the state and stocking them in this universal zone. Well, if you look where Lake Sarah falls on the spectrum of genetic footprint, you can see that it was very much different from the strains that we had been bringing from the northern part of the state down into the southern part of the state and stocking them. So this kind of, is kind of eyebrow raising to us is where did this strain come from? If you look at its kind of close relatives, it's pretty similar to other kind of water bodies that were from the lower Mississippi. So that's kind of what we were thinking, but we didn't know how it got there or why. So this kind of sprung out into, okay, where did this come from? And so Lauren took samples from a whole bunch of different water bodies in southern Minnesota, and some of those he took were from the Cannon River watershed. The Cannon River watershed is just south of the metro. It's mostly in Le Sueur, Rice County, Goodhue, Waseca, Le Sueur, so south of the metro about 30 to 45 miles. But it's a large sprawling watershed. And when he sampled those fish from this population, what he found was that there was a relatively high composition of this lower Mississippi River genetic strain that was existing in the Cannon River system. And that was especially the case if you look at the lower part here bounded by Lake Tatanka and upper and lower Cicada, where it comprised over just 60% to 90% of the population, which is relatively high, especially considering we hadn't been stocking the strain that we knew of. So that was kind of an important thing. Well, if we look back at history, they used to run Waterville Fisheries Office used to run an egg take station, kind of a small station here at the confluence or right above the confluence of the Cannon River to Lake Tatanka. And they took those eggs at that time and they would bring them across the lake to the hatchery. This is Waterville State Fish Hatchery's location, and they would incubate them. And then, at the time we didn't really know much about them. We just put them on the rack with everything else, and they'd get distributed throughout the state. On a bigger picture, if you think about this system, it all flows downstream into the Mississippi River. Billsby Dam was put in at one point or another in the past and blocked passage of fish from the Mississippi River up into the Cannon River system. So over time, this developed its own kind of unique genetic strain. Fish likely always, these unique fish always came out of the Mississippi River, came into this Cannon River watershed, but this was blocked once Billsby Dam was built for sure. And so they kind of became their kind of unique separated genetic strain. Well, the egg take station, this is actually a picture of it, circa about 1978 or 79. I didn't have a date on on this on this pictures, but I found these in the file. This is the egg take station. This was basically a makeshift trap that we set up. At the Cannon River, they would put this steel grates across the river, very heavy, very dangerous to put in. I wouldn't have my staff do it now, but there's a throat at the um, lower downstream side. So this is downstream, look, and it flows this way. So there'd be a hole here, an opening, a throat, and the fish would swim into here. And then at the upstream side, it was just graded off. So this made a trap essentially in the river. And the staff would come in every day during the spring and they would seine it with the seine and then they would pull out fish. And this is Gary Barnard, one of our old hatchery managers from Waterville State Fish Hatchery, likely holding up a 1979 circa LMS walleye that who knows, maybe the eggs or the fry that came from that particular fish started Lake Sarah. We don't know, but that's an LMS fish most likely. So that we pulled in some staff that had been retired, and we didn't really know. All the staff at Waterville were new at the time. We didn't know where they used to operate this trap, and we found out that it was located right here. This parcel right here is actually an aquatic management area operated by the DNR, and so it had easy access. And they used to install the trap every spring right here, and that's where they'd take eggs. Now this is the confluence with Lake Tatanka, Cannon River. And we know from electrofishing in recent years that there's a lot of walleyes that congregate in this stretch during the early spring. So it made sense that they were setting the trap here, but there's especially riffles that kind of are intermixed through here, but especially one down below this bridge where we know there's a lot of walleye spawning happening. So that became kind of an important point when we move forward here into the 2023 and beyond. They used to take the walleyes from the trap. They'd bring them over to the hatchery. This is one of our 1 acre ponds at Waterville State Fish Hatchery. They set up net pens there. They'd put the fish in there. Likely had one for ripe fish, one for green fish, and they would kind of move them about from day to day. And they'd take fish out and they'd spawn them right here on the shore of the pond, or they'd bring them inside the hatchery if the weather was inclement and spawn them in there. But that's how our operation was run from the early 1980s through early 1990s. Just as a side note, this guy right here is Dirk Peterson, who was one of our former fisheries chiefs. So Waterville has a long history of staff coming through and moving into other parts of the state, including chief of fisheries. If we look at the historic egg take at that Cannon River site, the early 1978, 79, and up till late 1980, we didn't have really good records because it was really just kind of really a grotesque egg take operation. But started in 1982 through 1992, you can see we took eggs at that site. And they did a pretty good job, especially initially. They were getting 20 million eggs, about 9 million fry, which is a pretty significant quantity. But if you look what happened over time, we started to see a reduction in the number of fry they were getting from this egg take station to the point where in 1990 they got zero. In 1991, they only had 233,000 fry. I mean, we're talking about spawning only 4 or 5 females in a year. So you might ask, well, why did you ever stop this egg take station? It was working. The reason we stopped it is because the numbers of eggs that we were getting and the fry we were getting from those eggs became so small that we could go up into northern egg take stations like Grand Rapids at Cutfoot or Pine River at Brainerd, and we could take this number of eggs in just a matter of a couple hours rather than over a week or 2 week period. So it really became a workload issue. And at the time, keep in mind, we didn't know about any type of unique genetics strains that existed. I wish we had. It might have changed what we did. But the fact of the matter is we stopped the station from taking eggs because it wasn't really efficient and effective relative to other egg take stations in the state. But now we know we have this unique genetic strain. We kind of know maybe where they originated from, the Cannon River system, and were brought over to Lake Sarah at one point or another, accidentally really. But we wanted to assess, knowing this strain exists and knowing what we should do with it are two different things. We wanted to assess what should we do. So we huddled together as a group of fisheries managers and staff that were interested in this topic and research. And we kind of came up with an idea knowing that we had an egg take station at Lake Sterra that was fairly successful for a few years. They were getting enough fry, 2 to 5, 6 million fry a year, that we could use and leverage those into some type of a research project that could help answer the question, should we be putting more time and more effort into this lower Mississippi River strain that we had identified that we didn't know about before? So we did that, and our geneticist and our research biologist, Dale Logston, who used to be stationed at Waterville, is now since retired. They came up with a good research project, and that research project really entailed taking fry from our northern egg take stations to our upper Mississippi River watershed and mixing and stocking them side by side with fry we were taking from the lower Mississippi River strain, which would have been from Lake Sterra at that time, and stocking them at equal amounts. And this is called the common garden or a paired stocking experiment. You stock them equal amounts. You can go out and sample the population at a later date and you can determine whether the proportions are equal, they're performing equally, or if they're not equal, is one performing better than another. So that was kind of the gist of the study that we set up at the time to try to assess whether this lower Mississippi River strain was actually performing better than other strains we had been stocking previously. In regard to the study lakes, we had select waters that we chose to study so we could have this be controlled. Wyndham, of course, was running the egg take station out at Lake Sterra. We wanted them to be a big part of the study. They were the initiators of it. So they selected 4 lakes that they thought would be good candidates for inclusion in the study, for this paired stocking study, and they had those 4 lakes. But then we also wanted to look at the impact of stocking Mississippi and lower Mississippi River strain on the Cannon River system itself. So we had core lakes we included in the study, Tatanka and lower and upper Cicada, and then also Cannon and Wells Lakes. So see, these were the core lakes included in the study along with the 4 in Wyndham. Because we're stocking both strains and we needed to maintain that proportionality throughout the system, and knowing that walleye fry can move in the system, we also had to include the upper or the headwater lakes in our study as well. So these got included by nature of we didn't want to screw up the proportions by stocking only Miss up here, only LMS, and having it mess up our proportions here. So we stocked the whole system with 50-50 LMS and Mississippi River strain. And then that's how our study was set up. So these were the study lakes. What we looked at was 4 general things. We looked at relative survival. What was the deviation in our population later on after stocking from 1 to 1, where 1 to 1 would equal survival. So that was an important factor. Is there a performance advantage by stocking LMS? We wanted to figure that out right at the top. What about relative growth? Does one strain perform better than another? This also can be important in how a strain performs long term, but also in determining which strain would be better to stock, Mississippi or LMS. We looked at relative cost. Just because one strain performs better, if it costs you $1 million in order to get 10 eggs, it's not going to probably do you much good because it's not practical to take those eggs. So we did look at cost to determine whether taking LMS eggs paid off in the long run as far as incorporated into our management plan moving forward. And the final thing we looked at was the relative reproduction. We knew now that there was some reproduction happening, natural reproduction I'm talking, in the Cannon River system, Lake Sterra, but we didn't know what the extent was. So we wanted to really kind of get a nail down what kind of magnitude of natural reproduction was happening in these systems. So that was a part of our study as well. To assess this, we used 2 different sampling techniques that are common to our fisheries. We used electrofishing. Our electrofishing was done at night during the fall, and that's the typical time we do electrofishing to assess our stocking success or natural reproduction, because what we're looking for is these YOY, which are called young of the year. Those will be fish that were either naturally hatched or that we stocked in the spring of that fall. So we go out, we stock in the spring, or they may be naturally produced. We go out in the fall and we electrofish and we catch those fish. That's we use for an index kind of their natural reproduction or our stocking success. When we did that, we also catch quite a few age 1 fish. These are fish that are just 1 year old. They're yearlings, and so we would also include that in our assessment as well. So electrofishing at night in the fall became a big part of our assessment. In order to assess the overall contribution to the population, we have to use kind of a longer term sampling method. And our gill nets are our standard sampling method that we use in Minnesota for sampling most of our fish populations. And so we use gill nets for that. And gill nets will sample anything from, you know, a 1-year-old fish all the way up to, you know, infinity, whatever age they are, because our gill nets are broken down into different size meshes, and that allows you to equally sample or randomly sample fish of various sizes. So these are the two sampling gears that we used. In order to assess whether we had stocked fish or not stocked fish, we had used two different techniques. Historically, we've used oxytetracycline, that's what OTC stands for, in order to mark our fish. Oxytetracycline is an antibiotic that's used in the agricultural field, but in fisheries field, we can actually immerse newly hatched fry, into a mixture of oxytetracycline, and it's uptaken into their bony structures. And when we take those bony structures later on and put them under ultraviolet light, we can actually see that oxytetracycline that was uptaken. So as you can imagine, we can take millions of fry at one time, put them into oxytetracycline solution, mark them, then go stock them, and we can use it as a mass marking, relatively inexpensive technique in order to monitor our stocking long term. And that way we can identify, we have two different of the same strain fish, we can determine whether one's stocked and one's wild by its OTC mark. We also incorporated a new technology. We talked about technology change and allowing us to assess our genetics for walleye. It's changed even more since the early 1980s, where we can use it for our advantage and other purposes. In this case, we used genetic parentage, is what we called that. And it's really quite simple. If you think about, in the hatchery, we take a male and we take a female and we put them into a pan. So usually it's 2 males and 2 females per pan. And we put them into a pan and we mix it together, and all the offspring that come from that have a unique genetic code. As long as we take the genetics off this male and these females that are into this pan, we can go back later on and sample these offspring, whether it's 1 year or 2 years or 10 years down the road even, and we can track it back to a unique combination of males and females that made up that pan of fish. It's a very powerful way that we can track our stocking efforts, and we incorporated it into this project. And with some of the data, some of the output that you'll see is from that unique genetic parentage technique. It's really cool stuff. So I'm going to look at some of the results briefly. And so the first thing we're going to look at was that relative survival. Was there one strain that performed better than another? This is a graph, and I'll explain it real quickly. We did this sampling from 2018-19, 2020 we were shut down because of COVID, and then 2021 and 22. So there was really 5 years of sampling. And this graph shows the first year of sampling, but because we sampled for 5 consecutive years, we had multiple age classes that we would have stocked in later years that would be a part of the sample. So when you see these 5 lines, there's fingerlings that you're stocking, age 1s, age 2s, age 3s, age 4s that we caught in 2019, 20, 21, and 22. So this first graph from 2018 will have multiple samples incorporated into it to kind of show you how the pattern carried through. What this is looking at is the percentage of lower Mississippi River strain in conjunction to all the fish that were sampled. If we were to expect in these graphs equal contribution of Mississippi River with lower Mississippi River, you'd expect all these results to follow this line. Equal contribution, 50-50, 1-to-1. If lower Mississippi had a higher contribution in our sample, they'd be above this line. And if Mississippi strain, the northern egg take strain, had a higher contribution, you would see the results end up below this line. So just keep that in mind as we look at these graphs. I'm also going to highlight in these graphs the Cannon River watershed lakes, because I'm going to talk a lot about those, mostly because I was from Waterville and I like to talk about those. But if you look at the graph, what you're going to generally see is that in this 2018 sample, most of these lines in Lake Tatanka and Sakata, or most of these dots, fall above this 50-50, which indicates that the LMS was returning back in our samples at a higher rate than the Mississippi, indicating that there was better stocking survival of those fish. In fact, in a lot of these samples, like in Lake Tatanka, you're seeing they're up in the 80 to 90%, up to 100% range. So almost all the samples that we were getting back were the lower Mississippi River strain that we stocked. Similar in Cannon, although maybe to a lesser degree in some years, but still most of the years we had relatively high contribution of lower Mississippi River strain. So that was on the Cannon River. What about in the Windham area, where we're way outside of the area of these kind of where these originated is? And Lake Okabena, again, we had high contribution of lower Mississippi River strain. And Kansas Lake, very high contribution of lower Mississippi River strain in 2018. But in Round and Bingham Lake, we had a higher contribution of Mississippi. Overall, what we were looking at is that lower Mississippi River strains seemed to perform very well based on this 2018 sample, but it was really lake dependent. The Cannon River chain of lakes, it performed really well. And some of the Windham lakes, it did also perform well, the lower Mississippi River strain. But in some of the other lakes, maybe Mississippi was doing okay as well. Now I'm going to show you 2021. I'm not going to show you every year because really all the data looks just like this. If you look across the board, no matter where the lake was, you're seeing 75 or greater percentage of our young of the year fingerlings and also age one fish are being composed of lower Mississippi River strain stocked fish, which indicates that they're based on this data, there was a pretty strong survival preference toward the lower Mississippi River strain relative to the strains we were bringing from up north, which indicates that lower Mississippi strains seem to be a better alternative when it comes to stocking in our lakes. And again, this pattern carried through throughout the rest of the study. So it was pretty consistent from year to year. Secondly, we looked at relative growth, and again, does one strain perform better than the other? Does it grow better, and does that result in the better survival? If we looked at the data, and this is a model, I'm not going to get into statistics, but basically what we found is overall, lower Mississippi River strain had about a 2 millimeter growth advantage over the Mississippi River strain. Now, it is a growth advantage, but I would say from a biological perspective, over a 10-inch or 9 to 10-inch fish that's a fingerling in the fall, 2 millimeters is probably not a biological significant difference, but nonetheless, LMS did have a slightly higher growth rate than the Mississippi River strain. Next, we looked at relative stocking cost, and this becomes important when we start to determine whether it makes sense to continue to put more effort into a certain strain or another. This is actually Dale Logsdon standing here, and he's mixing up a batch of oxytetracycline. He's got all of his PPE on, and this is our paired stocking. So this would have been all the Cannon River lakes, and so this was one lake, this row was another lake, this was another lake, and that's how we kept them separate. So one would be one strain and one would be the other strain. There's our paired stocking. So this graph shows you the cost difference between the two strains. And in this case, we put it in a percent of missed cost because we've been taking the upper Mississippi River eggs for a long time. We had a good idea what those costs were. So how did the LMS cost for obtaining fry compare to those Mississippi strain? And if you look at this, if they were equal, it would be at this line right here. If it's above, that means LMS was taking a lot more cost. In this case, pretty much across the board, the LMS was taking 150 to 200% more cost to procure the eggs than it did to take the Mississippi River strain. And if you've ever been to any of our egg take stations in the northern part of the state, you'll see that these are big operations. They're handling many, many fish a day, thousands of fish sometimes a day. Whereas Lake Sarah, we were handling just a handful of fish a day. So when you're deploying the same number of staff over the same number of hours and adding in the travel back and forth all the way from southern Minnesota all the way to the hatchery, all those things were gone into our fry costs. You can see why that would add to our costs when we're taking such fewer eggs. So if you just looked at it in this perspective, you say, well, maybe LMS doesn't make sense to keep doing because it costs so much to do. But if you add in the cost per return for a fingerling to the population, now we're talking about actually contribution to the fish population. You can see how this flips the script kind of a lot. If you look at that cost, because there's so much higher survival of our lower Mississippi River strain in these lakes, it was about 25% of the cost per return of fingerling to the fishery. So now it becomes a pretty significant advantage, even though it costs more to procure the eggs and to produce the fry. What you actually get after you stock those makes it is much more efficient if you're using the lower Mississippi River strain because their survival is so much higher. You can actually cut how many eggs you're taking because you can stock much fewer fry. So overall, the relative cost for the LMS per return of fingerling was much lower than the Mississippi by about a quarter. The last thing we looked at was natural reproduction. We didn't have a good idea what this was in some of our lakes at the time. This tub is one gill net that we picked in the fall from Cannon Lake. And this may give you an idea where this is heading, but these are all young of the year fish that were stocked that spring. These were harvested in probably September. So they were just a few months old and they were already sitting at about 9 to 10 inches long. So these are some beautiful fish. What these figures show, these darker shaded pie charts, these are going to be our stocked fish, and each strain is represented. And the clear one is going to be our wild fish. So these would be natural reproduction. What this showed us when we looked at these results, again, this is from 2018. We broke this down into each year. These are the age 0 fish that were either naturally produced or stocked in the spring that we collected in the fall as 8 or 10-inch fish. We had about an 8-to-1 ratio in Lake Tatanka between lower Mississippi and Mississippi for the fish that we stocked. That ratio was pretty consistent between 5-1 and 11-1 over all of our study lakes. That comparison or that proportion between LMS and Mississippi most of the time. It wasn't always the case. There was some situations in 2018 where we had a high contribution to Mississippi in Bingham Lake, which is down in the Windham area. But overall, LMS was outperforming by proportion the Mississippi River strain. The other thing to look at on here is we had a really high contribution of wild fish. And these were ones that didn't align with anything we stocked. These were naturally produced fish. So 91% in Lake Tatanka were naturally produced fish. 41%, I mean, there was pretty high percentage, which was kind of eye-opening to us. We didn't realize there was this much natural reproduction happening in our lakes. Of this natural reproduction, we could also determine which strain they were. And if we looked at that, it really was eye-opening because on Lake Tatanka, of these 91% of the fish that we caught that fall, 91% of them are wild, and most of them were lower Mississippi River strains. So LMS was contributing greatly to the population of Lake Tatanka and Lake Sakata. Same on Cannon Wells, although there was a higher contribution of stocking. Natural reproduction made up a big component, and lesser so in the lakes that were in the southwest Minnesota. But I think it's important to keep in mind some of these are winter kill prone lakes. They likely didn't have the brood source available to be able to continuously reproduce the LMS strain. So we'd get winter kills, we'd stock Mississippi back in them. Some LMS would persist, through those winter kills, but very low proportion. So we would probably expect a little bit less contribution of LMS in these lakes just because of that dynamic. This kind of carried throughout all the years, so I don't need to show all the years' data. But one thing I did want to point out is that this is Lake Tatanka data and Cannon Wells. And if you look at this, this is all the years of the survey or all the years of the study stacked on top of each other. You can see that there was really strong contribution of natural reproduction in Tatanka Sakata most years. In Cannon Wells, it's a little bit different. It seems like downstream further from Lake Tatanka and Sakata, stocking was much more important. And that's becomes important when it comes to how we're going to manage those fisheries moving forward. Because there's so much variability from year to year on natural reproduction, there's lots of reasons this can happen. Hydrology, water levels, wind direction, spring water temperature fluctuations, all these different factors can impact our natural reproduction success. But because it's very highly variable in the Lake Tatanka and Sakata system, we went to a low dose, kind of stocking a low amount of fish every year, so that in years where we didn't have a lot of natural reproduction, we were getting something in that system, but keeping it a low number so that in years we did have hyper reproduction, we weren't adding to a system already with a lot of fish, reducing their growth and causing problems with that population longer term. So this was one of the main reasons we went to annual stocking on Lake Tatanka and the Cannon River system using the 250 fry per littoral area, which was much lower than we had previously done. So the study conclusions were that there was better survival for the LMS strain. Almost across the board, it was surviving about 75% about 75% of the fish we were collecting in the fall that made up the population were LMS based on our stocking. LMS costs lower per return of fingerling, even though the fry costs more. It still made sense to pursue stocking them because they are much cheaper because we could stock less and their survival is better. Our stocking rates could be greatly reduced. Where we had been stocking 1,500 fry per littoral acre, we were now stocking 250 fry, possibly could even less. and still getting better results, which was really significant when it comes to how many eggs we have to take at our egg take stations, and also the operations of the hatcheries and how many eggs we had to incubate and take care of and all those other things. There obviously was meaningful natural reproduction occurring, especially in the Lake Tatanka cicada area, but in all lakes there was some natural reproduction was happening that really, up until this time, we didn't have a good finger or a good grasp of that kind of magnitude of natural reproduction in southern Minnesota. Again, why it was considered a universal stocking zone, we just didn't know this was happening. And the bottom line was Adaptive Management 101. Operational changes are underway, and we'll talk about some of those here in just a second. The bottom line is we kind of changed our approach to how we stock walleye in Minnesota. This was our 2 egg take stations leading up until last year, and this was kind of what we considered a new, maybe not a genetic conservation area, but a genetic management unit where we would be more considerate if we had the LMS eggs or fry available to stock those LMS fry into, rather than taking strains from northern Minnesota that maybe don't perform as well. Now, I think it's important here to point out that I'll talk about why this divides here, but these are unique to southern Minnesota in this watershed that's highlighted, this lower Mississippi River strain. Because we have genetic management units, we do not want to take this and put it into other parts of the state because we could mess up those genetics in other parts of the state by doing that. So really this LMS is intended to be in this watershed because that's where we know it performs well. And these genetic conservation units are important to make sure we don't spread those to other parts of the state where we don't know what consequences might be to introducing the genetic material into those parts of the state. So this kind of becomes important for a number of reasons moving forward. This is the spot, if you aren't familiar with it, that delineates the lower Mississippi River strain watershed from other parts of the state. And it's the St. Anthony Falls, which is in downtown Minneapolis. It's located here. And any fish can't get above this. This basically blocks up this upper Mississippi River. And that's what keeps it differentiated from the lower Mississippi River strain. But it's important to point out that the Minnesota River enters the Mississippi downstream of St. Anthony Falls, which means everything in the Minnesota River is part of the lower Mississippi River strain. But so does the St. Croix River, and that also has the confluence below St. Anthony Falls, which means this isn't just a southern Minnesota strain. This also does extend, this watershed extends up into the northern parts of Minnesota in the Hinckley area and the St. Croix River Basin, because that flows downstream to the Mississippi River below St. Anthony Falls, which ironically, the Knife Lake and also the Ann River had some egg take stations they dabbled in back in the 80s and 90s as well. So interestingly, we used to take these eggs back then not knowing that they were this unique strain. Some other things that we learned is that if you look at the Cannon River system, there's really 6 main kind of dams. They aren't all complete dams. Some of them are more often than not, but these are the dams that are located on this system. Billsby Dam, we know downstream, is a place that fish can't get above. When we were talking about during the study to the Lake City folks who manage Billsby Lake, we were talking about, well, they have all this natural reproduction in Billsby Lake, and it wasn't aligning with their stocking years, and they were getting natural reproduction. So it was like, what's going on here? They submitted some samples up to our geneticist, and he looked into this, and what we found out, because during the study, when we were stocking up here with this genetic parentage, we knew what fish we were stocking. We could track them. What was happening is fish were going all the way jumping these 5 dams, ending up in Lake Billsby. And what they thought was natural reproduction that was happening, because they were not fish they stocked, were fish that we were stocking in the upper watershed that were moving downstream. What we learned is that fish like to jump dams. And if you think about the natural history of walleye in the spring are going to swim up into upper watershed. They're going to find those natural spawning areas with riffles. They're going to lay their eggs. Those eggs are going to hatch. The fry are going to kind of slowly move their way back downstream and find more fruitful waters for feeding. So that kind of makes sense. The problem when you have dams, though, is once the fish go downstream, in many circumstances with these dams, they can't get back up. And that also has consequences for adult fish that might typically have been using this for spawning grounds that now can't get back up here under certain hydrologic conditions. So we found that out. Fish are moving downstream. The other thing is we don't When we think about this system, we don't really know where this natural reproduction is happening. We don't have a good handle on that. We suspect that a lot of it's happening in this area, given the contribution of LMS. We know there's a lot of reproduction that goes through here because we've run an egg take station here. But in general, we don't have an idea if there's a systemic type of contribution to the walleye population by spawning throughout the system, or if there's not. So that's something we really are thinking about, like what's the next steps in researching. That would be something we'd really want to look into, because maybe there's an opportunity to enhance some of the habitat what fish are using in these other locations, or even here where we know that they're spawning. So what does this mean playing out into the fish populations? Because that becomes important. Just because fish are stocked and they survive till fall, does that mean they're contributing to the population? If we look at our electrofishing data, which again is fall electrofishing, looking at young of the year fish that we were stocked or hatched that year, we can use this as an index of whether there's a strong year class likely or not. And if we look at these shaded boxes on Tatanka and Cannon, our years since we started stocking these lower Mississippi River back into the system, you can see we've had two of our highest points ever on Lake Tatanka for fall young of the year using electrofishing. And on Cannon wells, we're really seeing a huge response from our LMS stocking, where we're getting returns now in the fall that are just like, I mean, it's like night and day difference between now stocking LMS and when we were stocking Mississippi River strain. So from that perspective, it looks like it's making a difference. To assess whether it's contributing to the overall population, we have to use our gill netting. On Lake Tatanka, again, this is an index over time of our gill net catch numbers. On Lake Tatanka, we've had two of our highest catch rates since we started stocking LMS back into that system since 2018 when the study started. So our highest catch rates ever. And based on some of the angler information we're seeing coming back, we're going to sample this again in 2026. We're going to likely see pretty high numbers again because people are catching walleyes everywhere at rates really we haven't ever seen out there. So it seems that our stocking of LMS is really making a big difference. And the same thing on Cannon, we're seeing a high contribution, although not quite as significant as they are on the upper parts of the watershed. Some of our stocking has also shown a difference. This is Roberts Lake, which is not a main stem lake that you would see on the Cannon River, but it is connected to the Cannon River with an open channel. We were stocking Mississippi River fry in there since 2017, so 4 years of Mississippi River strain fry stocking. We got nothing. We started stocking LMS in 2021, and 3 out of the last 4 years stocking LMS fry, we've had a hit. So in places even where we had been stocking fry in the past, we didn't think they worked, so maybe we were doing fingerlings. We won't want to rethink that, because by putting LMS fry in some of those situations, it may work, may be much cheaper, and it may alleviate those fingerling needs for other purposes where they may be more needed. Same kind of thing on French Lake. A lot of years of Mississippi River strain. We didn't think LMS were working because we had a couple blanks, but then last year we got a hit. So we'll see how that continues moving forward. The bottom line is we have this new kind of genetic area for the LMS in this watershed below St. Anthony Falls. We now have 3 egg take stations to try to propagate eggs in order to accommodate our stocking needs in this zone. Those are at Lake Sarah, at the Cannon River there at Lake Tatanka, and then also Hinckley operated an egg take station the last 2 years. So now we're starting to build in a little bit of capacity for being able to stock LMS at a broader scale and be able to make it more sustainable. And that was really our goal even 4 years ago when we met and talked about this. What are the things we need? That was a source for these LMS fry so that we could stock them in a larger area. When we talk about stocking, if we were to stock the entire LMS watershed below St. Anthony Falls, it would take about 40 million walleye fry, which is a significant amount. And if we put that in perspective, just the Waterville area, before, it would take about 10 million fry to stock just those 9 counties in Waterville's area. That was at their old stocking rate of 1,000 or 1,500 fry per littoral acre. Because these perform so much better, we can stock them at a lower rate. And look what that does to our fry needs. In this case, it cuts it in half. And now if we look at this on an annualized basis, this is even years, odd years, and then combine in the ones that are done annually, we need about 3.6 to 3.7 million fry to accommodate Waterville's area stocking in the lower Mississippi River watershed each year. So that, and if we look at the historic egg take leading up to 2023 when we restarted the egg take station at Tatanka, we were almost accommodating that on an average basis. So that means it was something that, at least for Waterville, if they run the egg take station at Lake Tatanka, they could sustain their LMS needs from year to year um, through their own egg take. So that was really kind of something that said, let's do this. So in 2023, in the heart of the governor's fishing opener that was held in Waterville's area that year at Mankato, in the heart of Get Out More, pursuing funding for hatchery at Waterville, and all these things that were going on, we by accident started an egg take station again for the first time in 32 years. When Dale Logskin, our research biologist, was going out to test out his new boat in the river, this is the river in the right above the confluence, and he went up there to test his boat and said, "There's walleye all over." We went up there, we got our gear together, went up there, we collected some fish, and sure enough, we started taking walleye eggs again for the first time in 32 years at the Cannon River site. Ironically, at the same time, Fox News was there to do a story on the hatchery renovations. They ended up doing a story on this lower Mississippi River strain that actually played in nicely into kind of justifying why we need a hatchery at Waterville and why this funding allocation was so justified. So that kind of all fell together really nice. But that first year, we took about 60 quarts of eggs, ended up in 6.6 million fry. Again, we only need 3.4 to reach Waterville's goals. So we already did it that first year just by accident. Accidentally starting, but not by accident doing the work. So we did that all with electrofishing. Moving forward, we wanted to have a more refined method of taking eggs at this site. We are looking at a more refined like egg take trap, which would be a more traditional way for taking eggs. It's something you can deploy and go out and work, and it's more safer environment for doing that. Maybe you can work it in more conditions. This is the picture of the Boy River trap, and we basically purchased or modified that trap to meet the needs of the Cannon River, and that's the location we decided to put it, right in this spot here. So we actually purchased a trap in the fall of 2023. This was a picture of that trap. In that fall, we did a dry run installing it in the river. We picked that same spot pretty much again where we used to run the egg take station. And then the fall of 2024, we operated the trap. And then the very first day we had it set, we had a tree fall on it, so it wasn't a great start. But we had it operating that fall, and we did have some problems. There was so much filamentous algae in the river that year because we didn't have a lot of flow, and that we were setting this net. We were going and drop it in the evening, and within an hour or two, it was plugged with filamentous algae, and it was lifting the nets up here, and it was causing fish to swim underneath it. So we were having a little bit of problems with the design. We went out there one night and we staked this thing down. We held it down. You can see the algae that was built up in the lead, but we made sure that thing was fishing. And sure enough, that one night that we had that thing fishing, we caught 22 female walleyes. You can see their eyes all shining inside the trap. So we knew that was working. We know this is a good spot to do the egg take. So this is just a picture of our staff collecting those fish that we brought to the hatchery and spawned them out. Most of the time we're using electrofishing. We've done that again last year because we didn't have the modifications to our new trap yet. So we collected all of our fish in 2025 via electrofishing. It works, but water levels are pretty dictating that. Sometimes they can get too low for it to work. So we would like to have an alternate, more permanent solution to our egg take station. If we look at the historic egg take, both before 2023 when we restarted and after, we can see what our numbers were. What I wanted to point out was here, before 2023, we were getting about 3.4 million walleye fry a year, which would meet Waterville's quota. But since 2023, the 3 years we've operated, we're averaging 7.3 million fry. So we're getting enough fry now to not only meet Waterville's needs, but to meet the needs of several areas throughout the lower Mississippi River watershed zone. So really, this egg take is really adding a really big component to our long-term sustainability for lower Mississippi River strain propagation. We have a lot to learn. If you're watching the news last year, Waterville had a very, very serious flooding. When we went out the spring last year, this gauge of the river had to be pounded down in order for the water to touch it. By the time we were done in early May last year, the water started coming up. And by the time June came around, the water come up almost 4 feet, and it was over the top of our gauge at that time. This river is very dynamic. There's reasons there was years that back in the 80s and 90s they had years of zero because there was so much flow they probably couldn't set the trap. That's going to happen. We have to just make sure we have enough capacity built into our LMS egg take stations that we can pivot to other places to get the eggs that we need to meet our quotas. I'm going to skip some of this here, and that's the end of my presentation. If you have questions, I would appreciate them now because to be perfectly honest, we have a lot of questions too, and we still do, and this has taught us a lot from a fisheries management perspective. We are adapting as we go. We're learning new things, things we never understood before. We're trying to look at in the future what are things that would help us to learn more, like where are the walleye spawning in the Cannon River system, how are stocking these LMS into lakes and across southern, the LMS zone or the LMS watershed going to cause other lake seras to pop up that may be self-reproducing. Those are things we're still going to be learning moving forward. But this is a really cool story, and I'm really happy you got to tune in today to listen to it.
Greg, thanks so much for sharing all that. And it's true. You've probably heard me say it before at some of the fishing openers, like educating our youth and sharing this information. We're always learning something. We never have all the answers. So just keep on researching, keep on getting new people and new ideas in, and it's pays dividends hopefully in the future. So
For
it's
sure.
awesome. We got a couple questions in there, Scott. This is an easy one for me to answer. He's wondering if broadcast, if these are recorded. And of course they are. If you visit our website, mndnr.gov/discover, and look for the archives, you can go on there and you'll be able to find all the past recordings. And this one will be up probably Monday or Tuesday. So up there. Cassandra was asking, do you stock Lake Bilsby in Cannon Falls?
That's a good question for the Lake City folks, and that it was stocked in the past. I don't know if that's added to the LMS list at this time, but it does get stocked. They were including blank years in their stocking, and that's why they thought they had natural reproduction, because the years they weren't stocking, they were seeing those lower Mississippi or those fish show up. They didn't know their LMS. We tested them and found out that were moving downstream. So yes, it does get stocked, but I'd have to go check with the Lake City folks on whether it's getting stocked with LMS now versus other strains.
Good to know. I know a lot of those dams in the Cannon River, being a former Northfield kid myself, there's a flow over dams. So it's really easy for the fish to probably get downstream. It's the upstream travel, like you said, that is difficult.
Yeah. Just to put that in perspective, the dam at Morristown in 2000, I believe it was 2024, last year, 2 years ago, it was one of those 2 years. There was no flow over it. So the water was so droughty that there was no flow. But then in the spring, there could be multiple feet of flow over the top of it. So they're low head dams mostly. There are a couple that be pretty hard for fish to get above in Faribault. But most of them are low head dams where fish can go over, but they can't really get up in most conditions. So that's a really good point.
Right. You can stop your presentation if you want.
Okay.
William had one more question in here. Although I understand not wanting to stock LMS in northern lakes, would you expect that they would perform as well there, or are the lakes too different, deeper, colder, or whatnot, that you won't see as good of results?
Wait, that's a good question from multiple perspectives. One, we don't know, and that's always the issue, is we don't know. And that's why we have genetic conservation units set up in the state, is because it's important for us to maintain that genetic uniqueness of those specific areas. So one, we wouldn't want to do it because of that particular reason. And secondly, we don't know what the consequences would be, because they could perform very poorly, which is maybe likely or maybe not likely, or they could perform very well and mute out the native strain that's there and cause problems long term because of some other condition or habitat environmental stressor that we aren't aware of that could be present there that's not present in this part of the state. So those are kind of reasons we set up these genetic conservation units, so we can keep clear delineation of where it makes sense to put certain strains. So I would not recommend, and if people have called me and asked them this question before, I would not recommend taking lower Mississippi River strain walleye and putting them in parts of the states that they don't belong, that are outside of that native watershed, because we don't know what the consequences are to the native genetic strains that are there.
Yeah. We've had experience with the trout fisheries across the state, and Lake Superior strains of different trout species and stuff too. And I think you're correct on that. We don't want to mess around with Mother Nature too much. So, and we had one more question here. I think we got time for one more. Cassandra's asking, are the fish tagged that are being stocked? If so, how are you identifying them as stock versus natural reproduction?
Yeah, this gets back to the during the study, we were using oxytetracycline, which leaves the ring in any of their bony structures, like their otoliths that we take out of their inner ear. We were also using genetic parentage, which was the genetic tracking using unique genetic traits from the parents up for that pan of fish when we spawn them. Since that study was done, we have not been taking genetic material from them. So and we're not using OTC. So there's no way right now for us to assess natural versus stocked fish. But we plan, once we have the new hatchery at Waterville, right now we only have capacity to incubate one strain at Waterville without doing a whole bunch of other stuff that's really complicated. With the new hatchery that's being built, we're going to be able to separate eggs on that jar rack into unique strains, and then I'll also do small lots. So when we take our genetic material from fish, we can put that small lot on the jar rack, keep them separate, and we could just stock those, which means moving forward, we're hoping to be able to use genetic parentage to help answer some of those questions that you're alluding to for us to know whether it's stocked fish or not a stocked fish. But it really becomes a problem of we either have to take genetic material from all the fish that we're taking from all 3 egg take stations that get incubated at a site, or we take none right now given our hatchery design. But in the future, we'll be able to separate those out and allow us to have a little more capacity to do that at a smaller scale.
Great. Technology is an amazing thing. I'm glad to see we're putting it to good use and doing good things for the state of Minnesota and for everybody that likes to enjoy our fisheries across the state. So it's awesome. So Craig, I think we were just about out of time. Thank you so much for joining us. Everybody, next week we're moving from the water to the forest. We're talking about spruce budworm next week. And then we'll be back the following week talking about fisheries, the gar and the paddlefish, 2 species of fish that Craig probably is a little familiar with down in southern Minnesota. So a lot of
Absolutely.
Minnesota River and St. Croix River strains or fish areas where gar and paddlefish potentially are. So
yeah,
everybody get out, enjoy a fantastic weekend, take somebody new to the outdoors outside, show them what it's all about and have fun. Stay safe. We'll see you next week. Thanks, everyone.
thanks everybody.